It's been a week since I taught you something, since I released a video, and I'm sorry about that. I had a very hectic week in college this time. But yes, I'm here to teach you biotech applications. It is a very simple chapter. Simple as in some things are difficult to understand, but answering the questions in this chapter are very easy. And obviously the weightage of biotechnology principles is a lot more questions come from that, but biotech applications also has at least like 2-3 questions almost every year. So it's important for me to teach you that, and obviously if I teach it in continuation to the previous topic, it would be good. And yeah, it's a small chapter, it's an easy chapter, so we will be doing this today. And again, as I taught you before also, same format, I'll be teaching you, and then every around 10-15 minutes we will be doing a quiz. And because this chapter has a lot more like mug up that you have to do as compared to actually understanding, because the previous chapter was a very understanding heavy chapter. So I've put a lot more questions in the quiz, and we'll be focusing a lot more on questions, because even if I teach you, the only way you will actually remember it, retain it, will be through questions, right? So there are a lot of questions in the quiz. We'll be doing that also together, and as we always do, we have our PYQs. So we'll start with a revision and a few questions in the quiz every 10-15 minutes, and then we will end with the PYQs. Okay, so again, it's a simple chapter. It's just divided into 4 parts. You have agricultural, like the applications of biotechnology in agriculture, then in medicine. Then there's transgenic animals and ethical issues. Majority of the questions come from here and here, agriculture and medicine. So we can focus on that primarily. Transgenic animals and ethical issues are, I'll go through them quickly. Okay. So let me make I don't want to make the same mistake I made last time. I'll make myself small. Yeah. Okay. So we'll now start with agriculture. Yes, I just need to see if you can see my screen. Okay. Yeah. So again, biotechnology obviously has been very helpful in agriculture. It has been helpful in any biological science field, right? And so in agriculture, what is the purpose of agriculture? It is to ensure that there is food for everyone. It is to ensure that you get rid of the food scarcity as much as possible, and you're able to generate as much food as you can. So before in agriculture, we have been using agrochemicals. Oh, it's a very fat pen. Okay, so we have been using agrochemicals, right? And currently this is what our agriculture is very focused on using agrochemicals, on agrochemicals like pesticides and fertilizer. Then you have organic agriculture, and you have using genetically modified or genetically engineered organisms. So this is what our agriculture is primarily based on right now. And there are mainly in this chapter you have to remember the specific examples that help that like the questions come from. Okay, so I will not elaborate too much. So the green revolution is something we've all heard of, right? And the green revolution primarily focused on increasing crop yield by using better quality seeds and plants, by using fertilizer, insecticide, pesticide, and it did increase the food supply like 3 times, right? But the problem is there is only so much you can actually take from a particular piece of land. There is only so much you can extract for it, and like how much, even if everything is done perfectly, and you're giving your soil the adequate amount of fertilizer, you're using pesticides and all of that, it stops at some point of time, right? And that is what biotech helps us overcome. And along with that, one more problem is that the crops were improved, but not everyone can afford the agrochemicals and the pesticides and fertilizers, right? So one main important point in agriculture is tissue culture, right? So this is important. So tissue culture is also what they call micropropagation, the process of tissue culture. So I'll just write that down here. So this process name, the process of using tissue culture, is micropropagation. So what do? They have a whole plant. They already have Yeah, hello. Okay. So they have a whole plant and they take out a few cells of that plant, okay, and then they grow it in a test tube. So, first of all, what kind of cell are they taking? They are taking a cell which is called a totipotent cell. So, totipotent basically means a kind of cell, like, to give you an example, in humans, our zygotes are totipotent, which is like one cell which can give rise to all cells of, or all types of cells in that organism, right? So, that is a totipotent cell. So, what they do is they take a few cells from any growing part of a plant, okay, and they put those cells in test tubes in a sterile environment. They put it in test tube, sterile environment. They give it all the food that is required. They put a nutrient medium, right? Test tubes plus sterile environment plus nutrient medium. So nutrient medium will have everything. It'll have a carbon source. It'll have amino acids. It will have all the vitamins that are required. All the growth hormones that I already taught you about, right? So they put it in that and they kind of grow it in a test tube, and this now an entire plant can generate from this small extract of a plant which is called an X plant, okay, and then it will grow, and these are all going to be genetically similar to the plant that you've taken the cell out from, okay, so that means they are going to be soma clones. So, what does the word soma mean in biology? And you'll actually hear it a lot more. It's important for you to know. Soma basically means somatic. So, any cell that is not from a reproductive organ or it's not a reproductive cell, right? So, somatic is like, for example, if you have a somatic mutation in your body, there's like a mutation happening in your lungs, right? Now imagine that, yeah, as a smoker starts smoking and a mutation happens in their lungs and they get lung cancer. It's not their children would not get this mutation because it has happened in the lungs, in your lung cells. So that's a somatic mutation. But at the same time, if this mutation would happen in a sperm cell or an egg cell, then the progeny would also inherit the mutation, so that would come under the reproductive cells. So that is what a soma clone is, that it means that somatically it is completely similar to the cell that it was taken out from, okay, and is complete, and that is what somatic means, and a soma clone basically means that it is genetically identical to the original plant. And this has been done in tomato, banana, not very important, but yeah, okay, so that has been done. So one thing where biotech is used is in tissue culture, okay, then it's used for the recovery of a plant from, say, a diseased plant. So wait, I will shift this to a new page so I can put it all together. So basically what happens is that just give me a second. Yes, okay. So now you have pathogens that can infect everything, right? So naturally plants can also get infected. Now if plants get infected, then meristems, so what you have to remember over here is that the meristem, which is the actively dividing part, that never gets affected. So even if a plant is diseased, you can just take the cells from the meristem, make them grow again, and get a new plant. So again, the same way, this plant will be a soma clone of your original plant, but it won't be diseased. Fine. So that is one thing. So what is important over here, the only way they can ask a question from this is that to recover a plant from a diseased plant, where are the cells obtained from, and you just have to remember that they're obtained from the meristem. Okay, now I will increase my speed because I have reached 10 minutes, so I will finish the agricultural part, then we will do questions. Okay, now the next thing that they can use biotech is used in agriculture for is somatic hybridization. It's kind of funny only because I mean, this is that example of the tomato that they give you, which I always found very funny. Like, why, just why would you make that? They're both not very palatable vegetables individually also people find them difficult to have, and then you put them together and create a tomato. But anyway, so there's a somatic hybridization. Again, somatic means you are doing a hybridization, but you're not using the reproductive cells, you are using somatic cells. So how do they do that? They physically fuse them together, like they actually fuse the protoplast. So what is a protoplast? A protoplast is the living part of the cell, right? Like the cytoplasm is all the living part of the cell outside, and then you have the nucleoplasm. So whatever is the living part of the cell is the protoplast. So what they do is they digest the walls of two cells. Like imagine you take a somatic cell of a potato and you take a somatic cell of a tomato, and then you digest the cell wall, and then they kind of fuse them together to create a new plant. So that's somatic hybridization. So in this, the question that can come is either, of course, an example, which is tomato, again, which I find very funny, and what do they fuse? They fuse the protoplasts. Okay. Now finally, the most important part of agriculture is genetically modified organisms. So again, you have to genetically modify organism for some or the other reason, right? And what are the reasons that they use over here? I won't like write them down, but I'll just tell you. First of all, if you want to make your crops more resistant to pests, which is what we will be discussing in the BT, Bacillus thuringiensis, cotton, corn, and all of that. There's a lot of questions from that. So first one is, if you want to make it resistant to pests, you want to make your plants resistant to any sort of stresses, you want to increase the efficiency of mineral usage, right? So your plant absorbs a lot of minerals from the soil, but maybe it does not use all of them, and then you want to increase that efficiency, or you want to increase the nutritional value. So this one is important, increase nutritional value, because the example over here is golden rice. So a lot of people in India are vitamin A deficient, and that causes a lot of blindness problems. So we came up with golden rice, because rice is the staple of majority of the people over here. So if you put something in a staple that a lot of people eat, then they won't have a deficiency of it. Like another example of that was iodized salt. So you have golden rice, which is basically vitamin A enriched rice. Okay, this is important. That is to increase nutritional value. Now, to make plants pest resistant, we came up with BT cotton, BT corn, BT rice, tomato, potato, soya bean, all of these things. Okay, so BT basically stands for Bacillus thuringiensis. Yeah, okay. Bacillus thuringiensis. Okay, and how does this work? I'll keep it very simple. Bacillus thuringiensis is actually a bacteria that is lethal to a lot of insects. So what they did is they took the gene from the BT bacteria, which can kill a lot of insects, and they put that gene in your plant to make it create the same protein that is lethal to this thing. So it's not like the bacteria is growing in the plant or anything. There's just a gene. And the interesting part of your genetic code, the fact that it is universal, is what makes these kind of genetic modifications so easy. The fact that the genetic code in a bacteria would be expressed the same way in humans is actually very, very interesting and a very beautiful concept to understand, because that is what has caused so many developments and discoveries and inventions in I'm sorry, I'm yapping, but okay, I will go back. So you take the gene from Bacillus thuringiensis, which is responsible to create a lethal protein, and you put that in the plant. Okay, so now which all organisms are is this lethal to? So what they've given in the NCERT is lepidopterans, okay, coleopterans, and dipterans. Okay, so in lepidopterans you have the tobacco budworm and the armyworm, which sound like very fat insects. Then in coleopterans you have the beetles, and in dipterans you'll have flies and mosquitoes. Okay, so it's lethal to that. So what exactly it does is it creates a crystal, okay, a protein crystal, the bacteria, and this is created, very important, in an inactive form. So if something is inactive, we put a pro in front of it. Like inactive insulin is pro-insulin. So that way it's an inactive protoxin, and that is why it's not lethal to the bacteria that has it itself. And then what happens is this toxin goes into the gut of these this thing of the insects, and very important, in the mid gut. Okay, so in the mid gut, so if they write foregut or hindgut, it's wrong. It goes to the mid gut where there's an alkaline pH. Just write okay, alkaline pH. So that is where first of all these crystals get solubilized. Okay, then they become active, and once they become active, what do? They become active, and then they create holes in the epithelial cells of the midgut. Holes and pores in epithelial cells, which causes cell swelling and lysis. Okay, now what did scientists do? They took this Bt gene, they put it in the plant, and now the plant will be able to generate that protein. Now if this thing insect comes and it tries to eat that plant, what will happen? This protein crystal will enter that insect's body. What is its mechanism? It's inactive, it goes inside the body, and then in the mid gut, in an alkaline pH, it becomes soluble, and then it becomes active, it binds to the epithelial cells. Okay, and then after that it creates pores where there is cell swelling and there is lysis. Okay, so which all this thing examples of this I told you are cotton, corn, soya bean, right? And mainly to remember BT cotton, BT corn. Why? Because they've given. So which gene is it? Where do I write? I'll write here. Okay, so it's the cry gene, and very simple, it makes your plant cry. Okay, so there's two examples. You have cry 1 AB gene. Okay, and then no, I'll go with this thing. Cry 1 AC, cry 2 AB are for cotton. Okay, and cry 1 this thing AB is for corn borer. Okay, so cry 1 AC. So AC means C for cotton, but corn is also with C. Okay, so cry 1 AC and cry 2 AB are for cotton bollworms, and cry 2 AB and cry 1 AC are for cotton bollworms, and cry 1 AB is for corn borer. Okay, so these are the uses in this thing, agriculture. Okay, now one example of pest-resistant organism was Bacillus thuringiensis. Okay, now another example of pest-resistant organism is basically tobacco, but this thing that you need to know over here, the technology that you need to know over here is RNA interference. Okay, which I will take some time to explain to you, because I used to get very confused in 12th, because I feel like the NCERT had not properly actually explain what RNA interference is. Okay, so one pest resistant plant is Bacillus thuringiensis, and the other one is tobacco. Okay, fine. And so there is this nematode, Meloidecyne. Oh. Meloidecyne incognitia. Okay, again I forgot the yes, huh. So there's this nematode Meloidecyne incognitia which infects the roots, okay, of tobacco plant. So we obviously don't want that there, because that's going to decrease your yield and all of that. So what do? Is we do this, we kind of artificially induce this process called RNA interference. Now RNA interference, I will explain to you. So what the NCERT says is that there is complementary dsRNA that is introduced, okay, in the plant. So you have complementary dsRNA, okay, that will be made, and that will silence the mRNA of this thing nematode. Okay, so you guys already know that in genetics you have DNA that becomes RNA, and mRNA is basically your framework for how the proteins are laid out. So translation cannot happen if mRNA is not there. Okay, so what does the mRNA do? mRNA has to make the proteins, but if you silence this mRNA, then those proteins won't be made, so the organism won't be able to survive. Okay, and how do you silence the mRNA? By putting a complementary. So you put a complementary strand, which is, as you know, anything single-stranded genetic material um, is very lonely. It's looking for something complementary to join, right? And the moment another single-strand RNA comes, which is complementary to this mRNA, it combines, and then it's not able to get translated anymore because it becomes stable. The energy reduces and you cannot translate it at all. So that is what they do. They basically um, introduce a DNA into the tobacco plant. Now that is what is slightly confusing. First of all, it is dsRNA. So why is it joining? That is already double stranded. Why is it joining the mRNA? That used to be my first confusion. And second, I was like, who is fighting what? Where is the, how is the DNA going into the tobacco plant and then making the nematode unstable? So again, this is a concept that I'm explaining to you. If you do not find it important, you can skip it because it is a concept, and I'm telling you because I think it's important to understand. But questions के लिए तुम्हें यही याद रखना है कि complementary dsRNA होगा. Okay, fine. And obviously the name of the nematode and all of that, but RNA interference is used. It's found in eukaryotic organisms, and complementary dsRNA is used. Fine. But now to explain the process, which will that complementary dsRNA will silence the mRNA. But now to explain this process, what happens is in humans we have. I'll just put the process to this thing side. Okay. So in humans, we also can show RNA interference. What happens is a virus will infect us, for example, and if that virus has a dsRNA, right, our body instantly recognizes that this is not normal. The dsRNA should not be there. Okay, it is a foreign substance. Our body will recognize this as something that is abnormal and something we have to get rid of. So what it does is there's a protein called dicer which comes, and again, you don't remember this is just to explain the concept, and it basically snips this is the dsRNA of the virus. Okay, and this dicer will come and snip this RNA into small pieces. Okay, and these small pieces, siRNA we call them, it snips them into small pieces, and then it kind of takes and it splits them into small pieces, and it also splits them to make them a single strand. Okay, then it also makes them a single strand RNA which will get attached to an RISC. Not important, but basically this dicer comes, it sees the dsRNA, it snips it, and then it creates a single strand RNA which is complementary to the virus RNA. So when that mRNA comes to translate something, right, what our body has done is it has taken the RNA of the virus, made that single strand to fight the virus itself. Okay, and then it binds to that mRNA and it silences it. Okay, so this is what happens in humans. Now how does this work in the tobacco plant? Because tobacco plant में तो कोई immune system है नहीं. Basically over here the plant kind of acts as a virus to the nematode. Okay, so first what we do is we put the DNA in the plant. So we introduce a DNA, I'm saying DNA, using an Agrobacterium tumefaciens vector. Okay, so it's that TI plasmid, you know, right? So the tumor plasmid, we take that and we put DNA into the plant, and this DNA produces an RNA which is the RNA of a very important gene in the nematode. Okay, so then this produces, so the plant itself produces an RNA which is complementary to a very important RNA in the nematode, and this RNA that it produces is double stranded RNA. Okay, so now what happens is when the worm comes and the worm starts eating the roots of the tobacco plant, how do genetically engineered organisms work? All the cell have that certain modification, so all the cells now are producing this dsRNA randomly in the plant, which the plant does not need. But when the worm comes and it eats the roots of the plant, that dsRNA enters the worm's body, and the worm finds that dsRNA pathogenic. So what does it do? It tries to break it down, but that same dsRNA is actually complementary to the worm's mRNA, and then it binds to that. So it ends up killing itself in the process of eating this root. Okay, so that is how the RNA interference works. So again, this is a long story that I have given to explain it to you, but what is important over here to remember is that tobacco plant is the example, Meloidica and incognitia is the nematode, okay, and dsRNA, basically how it works is that dsRNA silences the mRNA of the nematode. And how is this done? DNA is introduced into the plant with an agrobacterial proliferation vector. That is what you have to remember, okay? So this puri kahani and all is not that important. So this is where it's used in agriculture. You have tissue culture, first of all, micropropagation. It happens in totipotent cells, okay, test tube, sterile environment, explants they are called, and they create soma clones, they're somatically identical. Then it is used in recovery of discarded plants, like that is basically tissue culture can also be used, right? Where you take cells from the meristem and you cultivate them. Then you have somatic hybridization, where protoplasts of cells are fused after you break down their cell wall. And then you have pest resistant plants increasing, then you have genetically modified organisms. Now in genetically modified organisms, examples that are important are, one is golden rice because it has vitamin A. Then you have pest resistant plants, which are, one is Bacillus thuringiensis, mechanism I explained to you, and then you have RNA interference also explained. Okay, so now let's do some questions in the quiz, guys. Okay, so these are questions from throughout the chapter, but it's okay, it will help revise everything, and then after that we will do its uses in medicine. Okay, it's mainly that agriculture and medicine, ethics and transgenic are a very small part. Okay, so let's do this question. Why is early detection of pathogen using conventional methods like serum or urine analysis often difficult? Okay, so serum or urine analysis are the tests that we standardly do in hospitals to check when somebody has a disease. It'll show up in your blood, it'll show up in your urine, but it only shows up in your blood or urine when the disease has progressed to a point where it is showing up in your blood or urine. So that is it happens very late. But we want diagnosis to be made as early as possible, because the quicker you treat a disease in its progress, the easier it is to handle, the lesser effects it has on the person's body. Okay, so yeah, so you will end up, so you want to make the diagnosis fast. If I teach you this now, I won't have to teach you this when I'm doing molecular diagnostics. Fine. So why is early detection important? Why is early detection of pathogen using conventional methods like serum or urine analysis difficult? Because by that time you can't do early detection, because the pathogen count itself is not high to show up in your blood or urine. Pathogen, so the answer is pathogen concentration is too low at early stages. Okay. The Second Amendment of the Indian Patents Bill takes into account which specific issues related to global bioresource exploitation. So, first of all, Indian Patents Bill Second Amendment was made, which took into account, patent issues like emergency provisions, research and development initiatives, and patent terms, right? So, okay, it's the fourth option, but this is what is given in your NCERT. So, in ethical issues, there are like only two main things that you have to remember, three main things, and in that one of them is this. What does the Second Amendment of the Indian Patents Bill Patents Bill deal with? Patent terms, emergency provisions, and R&D initiatives. Okay, what is the term used for bio resources by multinational companies without proper authorization or compensatory payment to the concerned people? Bioresources are being used by MNCs, okay, without giving them proper authorization or compensatory payment to the concerned people. Answer is biopiracy. This is just the basic definition. In 1997, an American company obtained patent rights for a new variety of basmati rice that was actually derived from what source? So this is what ethics comes into play, right? This is what is given in the ethics section, where what's happening is, like, there are certain traditional practices or diversity that already exists in India, and people in the West just pick that up, make a few modifications, and they claim it as their own, right? Something very often that happens very similarly with yoga as well. So an American basmati rice obtained patents rights for a new variety of basmati, okay, and as they've said, there are like 200,000 documented different types of varieties of basmati rice, and it's all given in your NCERT. But what did they do? That company basically just crossed Indian farmer varieties with, so they took the Indian farmer varieties, just crossed it with semi-dwarf varieties, which made the plants shorter, and they called it a new and improved version of rice. Transgenic mice are currently being developed to replace the use of which animal for testing the safety of polio vaccine batches? Tell me, what is the answer for this? The answer is, say if you can guess also, it will be monkeys because they are kind of the closest to us genetically. So transgenic mice are basically mice where you've changed the genes for certain purposes. I'll tell you what, but one of them is to make them, you can make them as similar in physiology to us, to humans as possible, because if you do that, then you can test all the testing that we do for medicines and cosmetics or chemicals. You can do that on the mice instead. Which organization was established by the Indian government to make decisions regarding the validity and safety of genetic this thing research? Genetic Engineering Approval Committee. That's it. Okay. Then, which human protein produced by transgenic animals is specifically mentioned as a treatment for emphysema? This you guys have to remember. You will do your MBBS and you will learn this. It's alpha 1 antitrypsin, very important. The first transgenic cow, Rosie, produced milk that was nutritionally superior for human babies because it contained which human protein? The answer to this is human alpha lactalbumin. Okay? Over 95% of all transgenic animals are which of the following species, guys? This is very basic. The answer to this is mice. Okay? Don't put rats accidentally, and don't think it's monkeys also. It's mice. We've also seen a lot of mice, small cute mice on campus they're fine only, they're not dying. Okay. So then let me take what is the primary principle behind enzyme linked immunosorbent assay? Antigen antibody reaction. Okay, I will not explain the mechanism to you, it's not important. You come to MBBS and you will learn it every year, you will learn it a lot, but it is antigen antibody reaction. In a molecular, and in this reaction you can detect the antigen that is present, or you can detect the antibody that your body creates against this antigen. Both you can detect. Okay, in a molecular diagnosis using a radioactive probe and okay, I will teach you more and then we will come back to these questions. Fine. So its uses in agriculture we covered. Now let's come to uses in medicine. Okay. So primarily it's used in medicine for recombinant therapeutics. Now what are recombinant therapeutics? Already told you, right? Recombinant means you've taken the DNA, put it in something else and created something out of it. So that is where this thing. So there are currently 30 recombinant therapeutics, okay, that are present, out of which 12 are being used in India, right? So 30 are approved, 12 are in India. Fine. So you create recombinant therapeutics. And the most important one from here, in which you get a lot of questions also, is insulin. Okay. Now what is insulin? Insulin is a very, very important hormone that your pancreas secretes. Okay. So it regulates your blood sugar levels, your blood glucose levels, and as you know, any problem or derangement in insulin production, in insulin sensitivity, causes diabetes, right? And diabetes is a problem that currently around 30% of the world's population has. India is the world's diabetic capital also, and when you will do your MBBS, you will understand that you will have to deal with so much diabetes. Every other patient you see is like, along with any issue that they have, they're also diabetic. Okay, so obviously when so much of the world's population is dealing with it, you need to be able to treat it. And one of the things that are important for the treatment of diabetes is insulin, right? And that is it's used in type 1 diabetes at a later stage, and type 2 diabetes, right? So what first they used to take insulin from cattle. I think I've zoomed in too much, no? Yeah, it's like so big. Okay, cattle and pigs. But this creates a lot of problem, primarily immunological responses, right? Like our body can often have a lot of immune responses to the insulin extracted from cattle and pigs. So what did they do? Eli Lilly, again, recombinant therapeutics, they artificially created insulin. But there was a problem with this. So I will explain that to you. So insulin is in humans naturally, okay, in us, it exists in 3 chains, okay? It exists like this. Fine. So you have your A chain and B chain and C chain, and this is proinsulin, right? But what you need is not pro is like the early stage. What you need, these are like, so you have these three polypeptides, but you need active insulin, which is A chain. Okay, A chain, B chain, linked by disulfide bonds. Okay, this is what we needed, because what will we do with proinsulin? We don't need proinsulin, right? Like, I mean, we need active insulin more than we need proinsulin artificially. So then the problem was, okay, we can take the genes of insulin, we could have put them in a bacteria, in E. coli, like Eli Lilly did, and then the problem that would have created is it would have made A, B, and C polypeptide. So what did they do? Okay, in 1983, Eli Lilly, very important. What they did was, they created A and B chains separately in E. coli. Okay, in E. coli, plasmids they put in this thing, plasmids of E. coli, they created it separately, then they extracted them, and then they linked them with disulfide, guys, very important. Okay, disulfide bonds, and then they found human insulin. And this made not only the process of making insulin, obviously it's so much easier to make insulin like this as compared to, extracting it from the pancreas of a pig and cattle and then taking it, and then this made the process a lot easier. So this is what Eli Lilly did. Okay, so first example that in medicine is insulin. Next example is gene therapy. Gene therapy, basically, see, there are some diseases that are genetic diseases that exist because there is a defect, an intrinsic defect in the gene of a person, right? And these are, again, they can be inherited, they can happen because of mutations that happen de novo, but it is genes that are inherited that can cause these genetic diseases. And the best way, almost always, the best way to treat something is to fix the cause, right? So then scientists thought that, okay, if the cause of a lot of the diseases is a genetic defect, let us try fixing the genetic defect, right? But how do you fix a genetic defect? Okay, imagine I have a genetic defect. I have so many cells in my body. How would you fix it? Okay, think about it for some time and you will understand that you will have to change the DNA of all my cells, every single one of my cells. Now the thing is that you can, again, in theory you can say that, but even once you change the DNA of all my cells, okay, now imagine you change the DNA of my entire skin, okay, currently. So somehow I had some disease where there was a lack of an enzyme, and now that my body, you have somehow managed to do it, will start making that new enzyme. But now my cells will die. The new cells will come. So those won't have the modification that you had made, right? So that is what is very important, that you have to, scientists are trying to figure out how to do gene therapy in a sustainable way such that it exists in all the cells and it can, actually be sustained throughout a person's life. Anyway, so it is a gene therapy. It happens in, you have to do it to treat a genetic defect. So what you remember over here is, first of all, in 1990 was the first instance. A 4-year-old girl had an ADA deficiency. Adenosine deaminase deficiency she had, okay? And this normally causes an immune linked disorder, like your immune system is not very weak if you have an ADA deficiency, okay? So normally, what are the modalities of treatment that we have for an ADA deficiency currently? We can add a bone marrow transplantation, right? Or we can give the ADA enzyme artificially. Now, if I'll explain, why would you do a bone marrow transplantation? So an ADA deficiency is basically when our body is not creating ADA, and that I told you is very important for immune cells. And immune cells are made in your bone marrow, right? So if you put new bone marrow in a person, and I told you there are stem cells, like you guys know that there are stem cells in a body, right? Cells that can give rise to other type of cells. So essentially, like the higher up you go in the hierarchy of cells, at the stem cell level is where the issue is, because those cells are not producing cells which have ADA. So if you replace someone's bone marrow, put somebody else's bone marrow, somebody who can create ADA, then you will have lymphocytes which will have this enzyme in it, right? And it won't be a problem. So that is those are the modalities of treatment that are present. So what these patients for currently, now what scientists did for this girl was that they took her lymphocytes only, okay, and they artificially inserted functional DNA. So they artificially functional ADA DNA. So now ADA is not being produced because of some issue. Like imagine your DNA doesn't have the gene that produces ADA. Okay. So what did they do? They took the lymphocytes of this girl. So now that they're taking the lymphocytes of this girl, so obviously rejection of this lymphocyte or any immune response to this lymphocytes will not be an issue because they're from that girl only. And then they put the ADA DNA in it through a retroviral virus. Retrovirus basically. Retroviral vector. And they transformed the cells, all the lymphocyte cells of this girl, and then they put these lymphocytes back into her. Okay, so now these new lymphocytes had ADA, and that is how they managed to, for the first time ever, treat the ADA deficiency. But the problem is that lymphocytes are not immortal, right? So technically they still didn't treat it. You still need periodic infusions of lymphocytes, but it is a step towards a towards genetic therapy. But think about it theoretically. If you had to treat the disease, how would you do this? If you know that you have a child who has ADA deficiency, how would you ensure that the ADA deficiency doesn't manifest when the person grows up? And what you would do for that is, when the human is in an embryo stage only, you would genetically try to modify the embryo. Only then all cells would always have ADA as the person would grow up, and that would be a permanent solution. But that is also very difficult to feel, like that's not very feasible. Okay, so that is one use, gene therapy. Then you have molecular diagnosis. So now this is something I already taught you, but while we were discussing questions, but serum urine analysis, right? This is your standard modalities of analysis for a disease. They are this thing in the later stage when the pathogen count is high. For an early diagnosis, we need something that can pick up your pathogen at a very small concentration only, right? Because what happens, a virus or a bacteria, once it enters your body, it multiplies, and you want to be able to control it before it multiplies. And what do we use for that? For an early stage, we use things like PCR and ELISA. Okay, so what did a PCR, you already know. What does it do? It basically takes the DNA and it amplifies it. Okay, if you have one virus, like a single virus, which has nuclear, which will obviously have nuclear material, so PCR would amplify it so much that you'll be able to pick it up at an early stage also before the virus has spread. And ELISA works at an antigen antibody reaction, and it can detect the antigen and the antibody. It's not like it's just detecting the antigen. Okay, antibody matlab the antibody we create against that antigen. Fine. So these are the two modalities for early stage diagnosis. Then you want to be able to detect mutations also. Okay, now how do you detect if you want to detect mutations in very specific sequences in this thing, in your genetic material? So how would you detect? So we use molecular diagnostics to detect mutations as well. Okay, now how do you do this? Is basically you work on the complementarity phenomenon of this thing only, like of DNA, like how in RNA interference no that single stranded RNA was there, then single stranded complementary came and bind. So what we do over here is we take a person's DNA and we make it single stranded, and then we add a complementary DNA. So now for example, I know what normally human DNA should be like. Okay, now imagine I'm taking a segment of genes that has, again, let's say genes related on some chromosome related to some related to your lungs function, right? So that's a very simple and like simple way to put it. But you've taken that part of the DNA, then what do? You make that DNA single stranded, and then you apply a complementary anything single stranded DNA or RNA, right? Single strand, and you make that bind. This can be done on RNA, it can be done on DNA. You make that bind with the DNA of the person that you are investigating, right? Now, what is special about this complementary DNA or RNA is that it is radio labeled. Okay, it is labeled with a radioactive it'll be labeled with radioactive nucleotides. Okay, so why is that important? Because radioactive nucleotides will show color. Like, you can't randomly just take DNA, separate it and look at it, right? Even when you do gel electrophoresis, you have to stay in it and look at it under UV light. Same way over here, I take a human's DNA, okay, I attach a complementary DNA to it, which is radio labeled, and then if it shows color, that means if you can actually visualize the complementary DNA, that means there are no mutations, okay? But whichever areas have mutations, this complementary DNA, this complementary probe, so this complementary DNA that hybridizes it is called a probe. So this complementary probe will not be able to bind to the area that has a mutation, and because of that, no color will be emitted, right? So it will not appear on a photographic film. Guys, did you understand this? This is also important. Questions come from this as well. Okay, so in molecular diagnostics, we can this thing. You take a labeled you take first you have PCR and ELISA that can be done, okay, for early detection. Then you have when you have to detect a mutation, you have to take complementary DNA, attach it to the person's DNA, and if it is complementary, that means there's no mutation, it will bind and it will appear on a photographic film. But if there is a mutation, you will not be able to see it, okay? So these are the basics that you have to understand. But very, very important that if there's a mutation, there is no complementarity, so you will not be able to see it, okay? So these are its uses. These are some of the uses of biotech and medicine. There are so many more, okay? And to revise all of this: medicine, insulin, gene therapy, molecular diagnostics, okay? Now let's move on to this thing. Transgenic animals. You want to do transgenic animals or you want to do the quiz now? Let's do the quiz, okay? I've been talking for a lot of time and you would have gotten bored, but after that we move on to, it's very small, okay? Transgenic animals and ethical issues. I will not take more than 5-10 minutes. So let's just do the quiz for now. Fine, more questions in the quiz. We've completed like half, okay, kind of half of it. Fine. So in molecular diagnosis using a radioactive probe and autoradiography, why does a clone having a mutated gene not appear on the photographic film? Okay, because mutated gene will not be able to bind with the radioactive probe. Why? Because it does not have complementarity. So the answer is the probe lacks complementarity with the mutated gene. Okay. Why is the polymerase chain reaction useful for the early diagnosis of diseases like AIDS or cancer? So in these kind of diseases, it's very, very important, right? Because you want to pick up on AIDS or cancer before only. Answer is because it amplifies very low concentration of pathogen nucleic acids, okay? What is considered a potentially permanent cure for ADA deficiency according to the NCERT book, okay? This, I already told you, introduction of the ADA gene into cells at early embryonic stages. In the first clinical gene therapy trial in 1990, 4-year-old girl, why did the patient require periodic infusions of genetically engineered lymphocytes? Why did she require? Because as I told you, lymphocytes are not immortal, so they die after some time. So once they die, all lymphocytes have a different life. Then you need new lymphocytes. So that is why. How did the company Eli Lilly successfully produce mature human insulin in 1983? A chain बनाया, B chain बनाया, plasmid में डाला, E. coli में डाला. They made 2 chains separately, then they joined them with disulfide bond. So answer is D. They produced A and B chains separately and combined them. What was the main challenge in using rDNA techniques to produce mature human insulin? What was the main challenge in using recombinant DNA to how would you get it in a mature form without the C peptide. So don't get confused with option D, because we do not want C peptide. The answer is getting insulin assembled into a mature form. Why are recombinant therapeutics generally considered safer than similar products isolated from non-human sources? Why? I told you, because there's immunological reactions where a lot of people can have anaphylactic shocks because of an immune response to any sort of foreign antigen. In the process of RNA interference, what could be a potential source of the complementary RNA that initiates silencing? Okay, so this is given in NCERT, that transposomes that replicate via an RNA intermediate. Okay, so transposomes are jumping mobile genetic elements. So they will, if they replicate using an RNA intermediate, that could help in silencing the mRNA, this thing. RNA interference involves the silencing of specific mRNA due to the formation of which molecule? Answer is dsRNA. Please remember, it's not ssDNA, dsRNA. Which specific cry gene is mentioned in the text being used to control the corn borer? Only one gene is there, so it's cry1AB. Okay, so because there's only one gene, there's cry1AB. Cotton, this thing, bollworms have two genes, so it's cry1AC and cry2AB. The specific activation of Bt toxin in the insect gut is triggered by which environmental factor? Alkaline pH of the gut, guys, important. Why does the insecticidal protein produced by Bacillus thuringiensis not harm the bacterium itself? Because the toxin exists as an inactive protoxin. According to the source material, which modification is specifically cited as a way to prevent the early exhaustion of soil fertility? I told you you don't want the soil fertility to get exhausted early, right? So you want to be able to increase the efficiency कि देख जो minerals उन्होंने लिए उनको भी ढंग से use कर पाए. So increasing efficiency of mineral usage by plants. In agarose gel electrophoresis, why do smaller DNA fragments migrate further? This is from last chapter. This one you don't want. Which part of an infected plant is typically utilized to obtain virus-free progeny because it remains unaffected? What did I tell you? What do you have to remember here? The meristems. What property of plant cells allows for regeneration of a whole plant from an ex-plant under sterile conditions? Answer is totipotency, not to be confused with pluripotency. So I'll tell you the difference between the two. So the difference between totipotent and pluripotent is very important, and that is that both are types of cells that can give rise to lots of types of cells. But totipotent is like remember it at total potent. Then it can give rise to all the cells that are present in an organism. But pluripotent can give rise to a lot of cells, but not all. So the answer is because you want the full plant, right? So you'd want totipotent only. You would not want pluripotent. Okay. So this is the quiz. Now I'll move on to transgenic animals and ethical issues very quickly, and then we will do PYQs. Okay. So transgenic animals is any animal. Wait. Yes. Okay. So transgenic animal is any animal. Oh, it's green in color. I don't like it. Okay, animals with modified DNA basically. And 95% of them are mice. Okay. So why do we need transgenic animal? What are the uses of them? Okay, so first of all, just to study normal physiology and development. It's not just you want to do something special, but to be able to create any discovery or do anything in a body, you have to be able to study its normal physiology as well. So just for that, if you can introduce genes that are, in humans, in mice, and to study how they work, then that's what you need this thing for, transgenic animals for. Okay, then to study the disease, the progression of a disease, you need them. Then, and there are a lot of transgenic models that exist for cancer, for cystic fibrosis, right? I'll write this down only. Okay. So uses are normal physiology. Then you need it for diseases. So there are already transgenic models that exist to study cystic fibrosis, cancer, rheumatoid arthritis. Okay, these exist. Then if you want to develop some biological products, biological I was writing. Yeah. Biological products. Okay, so if you want to obtain some sort of biological products, now example for that is given is alpha-1 antitrypsin. Okay, just remember it because you will learn it when you come to MBBS. Alpha-1 antitrypsin to treat emphysema, very important. okay, to treat emphysema. And another important example was in 1997, they made the first transgenic cow, okay? Very funny, but they named her Rosie. And what was special about her was that her milk had extra, this thing, um, grams of protein. It had extra human alpha lactalbumin. Okay, and how much extra did it have? 2.4 grams per liter. It's just, remember, if you're a bodybuilder, it's funny to remember, okay? Then you can use this thing, transgenic animals, to test vaccine safety. Currently, if you can make transgenic models which are more human, like they're more similar to humans, then you can do it on mice instead of monkeys. And for chemical safety, vaccine safety. To test vaccine safety and for chemical safety testing as well. Now coming to the ethical concerns over here, which is the fourth part of the chapter. Again, not a lot of questions from here. What all do you need to remember? First, that we have a genetic GEAC, Genetic Engineering Approval Committee, because there's a lot of ethical concerns with genetically modified organisms. So that is one thing you have to remember. So second, remember definition of biopiracy, which I already taught you, is basically when MNCs use, certain traditional processes without the authorization from those countries. Example of that was given in as basmati. Again, we already discussed that in the questions. And remember that the Indian Patents Bill had a second amendment to discuss all those emergency provisions and research and development initiatives. Okay, this is all that there is to remember in the ethical parts. So this is the chapter. You have its uses, biotechnologies, applications in agriculture. Those are very important, especially BT cotton and this thing, BT corn and RNA interference. Then in medicine, insulin has a lot of questions. Gene therapy has a few too. Molecular diagnostics I taught you. And then you have transgenic animals. Again, very few questions come from here. Mainly you have to study agriculture and medicine properly. Okay, so now we can move on to the previous year questions. So the first question is, which of the following genetically engineered organisms, I zoomed in too much, okay, was used by Eli Lilly to prepare human insulin? Okay, so I told you it was prepared in, it was prepared in the plasmid of E. coli, right? And that is a bacterium. So the answer is bacterium. Okay. Next question. Following are the steps involved in action of toxin in BT cotton. Okay, so these are the steps, right? First option is the inactive toxin converted into active form due to alkaline pH of gut insect. Okay, that happens once it goes in. Bacillus thuringiensis produces crystals with toxic insecticidal proteins. Okay, the alkaline pH solubilizes the crystals. The activated toxin binds to the surface of the midgut cells, creates spores, and causes death of the insect. The toxin proteins exist as inactive protoxins in bacteria. Okay, so this has to be the last step. At least that's how I would do it. So D has to be the last step. Okay, D is the last step in multiple options. Then Bacillus thuringiensis produces crystals, right? I think this is the first step because the production of crystals has to happen first. Then the toxin proteins exist as inactive protoxins. This should be last. Okay, this should be fifth, right? So now third and fourth. Then third would be the alkaline pH solubilizes the crystals, and the fourth would be the inactive toxin gets converted into active form because of this. Okay, so the answer should be B, E, C. Sorry about the noise, guys. There is construction happening in my hostel, but the answer should be B, E, C, A, D. B, E, C, A, D. Right? So the answer is D. Yes. Okay, good one. Next one. Okay, yes. What is the fate of a piece of DNA carrying only gene of interest which is transferred into an alien organism? The piece of DNA would be able to multiply itself independently in the progeny cells of the organism. Not always. Let's see. Um, it is only able to do that if it is integrated into the genome. It may get integrated into the genome of the recipient, correct? May. It may multiply and be inherited, correct? The alien piece of DNA is not an integral part of the chromosome. If it is not integrated, then yes, it is not an integral part of the chromosome. It shows the ability to replicate, not independently, only if it enters. So, okay, so B and C is an option, right? So the answer is B and C. Let's look at what it says. Yeah, answer is C. Okay. Now, which of the following are fused in somatic hybridization involving two varieties of plants? I told you somatic hybridization, what do? You take the living part of the cell, you break down the cell wall, and you fuse it together. And that is, what is the living part of the cell? The protoplast. Yeah, answer is C. Okay. Okay, match the following. Alpha 1 antitrypsin, emphysema. Okay. Cry1AB, Cry1AC. So Cry1AB is for corn borer. I told you Cry1AC is for cotton and Cry2AB is also for cotton, but Cry1AB is for corn borer. So this is 4. Cry1AC is for cotton. Enzyme replacement is for ADA. So 3, 4, 1, 2. 3, 4, 1, 2. This should be the answer. Yeah, answer is C. Okay. So remember the genes Cry1AB and is for corn borer. Okay. Which of the following statements is incorrect? Okay, this is about bioreactors. This is from the previous chapter, but we'll still do it. A bioreactor provides optimal growth conditions for achieving the desired product. That is correct. Most commonly used bioreactors are the stirring type. That is also correct. Bioreactors are used to produce small-scale bacterial cultures. This is wrong. So this is the answer. They've asked for incorrect statement. Okay. So the answer is C. Now, next question. With the following diagram showing restriction sites in E. coli. Okay, this is also previous chapter, but we can do the question. Actually, this is a good question. We should do it. The following diagram shows restriction sites in E. coli cloning vector PBR322. Find the role of X and Y genes. Okay. So if you remember, for this you have to remember the diagram really, really well. And if you remember, X was your OREC and the origin of replication, which is responsible for controlling the copy number, and Y was, it was written ROP when the function of that gene is not given, but it is actually for the replication of plasmid, if I'm not wrong. So let's look at the options. Gene X is responsible for resistance to antibiotics? No. Which one is this thing? These two are responsible for resistance to antibiotics. Gene X is responsible for controlling the copy number. That is correct. And gene Y is for protein involving the replication of plasmid. That is correct. But you don't know the second part, but you know the first part, right? And no other option has the copy number, this thing, answer. So it has to be B, even if you don't know the Y gene. Correct. Okay, which of the following statements is incorrect? Okay, again, this was a very common mistake that we used to make, sometimes reading incorrect and correct. It is used to deliver incorrect about Agrobacterium tumefaciens. It is used to deliver gene of interest in both prokaryotic and eukaryotic. Wrong, is only for eukaryotic. So, that we got the answer. But let's look at the other options as well. TI plasmid from Agrobacterium tumefaciens used for gene transfer is not pathogenic to plant cells. It's called tumor inducing. Okay, it transforms normal plant cells into tumor cells. Yes, it's true. It delivers tDNA. Yes, so A is the answer. Okay. Yeah. Now, which of the following can act as molecular scissors? Very easy. Restriction enzyme. Let's do this. Match the following. Match the followings are my favorite questions. It just made sense. It was like very relieving when they would come, because even if you don't know half of it, you can figure out the rest somehow. Gene therapy allows correction of a gene defect. A is 4. RNA interference, cellular defense. 3. ELISA, diagnostic test for AIDS. Yeah, I mean, yes, can be. And electrophoresis is 1. So 4, 3, 2, 1. Ulta. 4, 3, 2, 1. Answer is C. Yay. Which of the following techniques does not serve the purpose of early diagnosis? Early diagnosis which does not serve the purpose of early diagnosis of a disease for its treatment is serum urine analysis. Very important. And I taught you also that serum and urine analysis are only done in the later stages. Okay. I mean, they're more helpful at the later stages. Milk of transgenic cow Rosie was nutritionally more balanced product for human babies than natural cow milk because it contained human alpha lactalbumin. C. Transposons can be used in which of the following? Gene silencing. Transposons are mobile genetic elements and they're used in gene silencing using RNA interference. Okay, let's do a few more questions. Okay. In gene therapy of ADA deficiency, the patient requires periodic infusions of genetically engineered lymphocytes because this one we already did. I already taught you because they are not immortal cells. So that is option D. So you can see it now, right? Yeah. Option D, genetically engineered lymphocytes are not immortal cells. Insulin we've already discussed. Okay, you can't see option D. D is safety testing, okay? When gene targeting involving gene amplification is attempted in an individual's tissue to treat disease, it is known as. Very simple, guys. Gene therapy. Okay. Insulin. Okay, fine. We'll do one insulin question. It's just we've explained it so much. Okay. With regard to insulin, choose correct option. Correct options. Okay. C peptide is not present in mature insulin. Correct. It's not present. The insulin produced by our DNA has C peptide. No. Why will you produce C peptide by our DNA? Proinsulin has C peptide. Correct. A peptide and B peptide of insulin are integrated by disulfide bridges. Correct. A, C, and D. That's an option. I got scared first. I thought A, C, and D was not. Oh, so you can't see it. A, C, and D is an option. Okay. See, answer is C. Then, okay, match the following. Bt cotton, Bacillus thuringiensis. A is 4. There's only one option. But let's look at the rest. ADA deficiency gene therapy. RNA interference, cellular defense. PCR is detection of HIV. So answer is D. Okay. Which of the following statements is not correct? Proinsulin has an extra peptide called C peptide. Correct. Incorrect statement. Not correct, Devas. The functional insulin has A and B chains linked together by hydrogen bonds. Wrong. It's disulfide bonds. Very important. Okay. You want to do more questions? Okay, we will do more questions. Yes. In RNA interference, the genes are silenced using, very important, dsRNA. Do not get confused. DNA is introduced into the plant by Agrobacterium tumefaciens vector, but RNA interference technology itself uses dsRNA. Okay? B. Which of the following is true for golden rice? Very important. It is rice that is enriched, right? Because a lot of people suffer from deficiencies, and it is a vitamin A enriched rice with the gene from daffodil. Don't worry about that. You just have to know that it is a vitamin A enriched rice. Okay? So yeah, that's it, guys. We've covered biotech applications. Told you very simple chapter, very easy chapter, and thank you for listening. After this, I will be doing cell cycle and division. And whatever else you need, please do comment. Please do drop your feedback and please subscribe to this channel. Thank you.